Good morning, my dear students. Today we are discussing the life history and pathogenicity of Entamoeba histolytica. So, this is the continued part of parasitology. So, coming to the introduction of Entamoeba histolytica was first of all reported by a Russian zoologist Losh in 1875 as amoeba coli from the feces of a patient suffering from dysentery. The species Entamoeba histolytica was established by Scardian in 1903 and he differentiated the pathogenic and non-pathogenic types. Krayag in 1962 has estimated that more than 10% of world population is suffering from the infection of this parasite. So this Entamoeba histolytica is worldwide, that means cosmopolitan in distribution, but it is commonly found in epidemic form in tropical and subtropical regions than in the temperate region. So here is the picture, the microscopic view of Entamoeba histolytica. So next, coming to the morphology of Entamoeba histolytica, you already know that it is an protozoan parasite. So this is an microscopic and lives as an endoparasite in the upper part of the large intestine, that is colon of man. So Entamoeba histolytica is a protozoan parasite that accounts for an estimated 1 lakh annual deaths. So it inhabits the mucus and submucous layer of the large intestine. It feeds mainly on the tissue of the intestinal wall and often produces severe ulcer and abscesses. Infection ranges from asymptomatic colonization of a large bowel to severe invasive intestinal and extra-intestinal diseases. In chronic cases, that means in severe cases, it may enter the blood circulation to reach the liver, lungs, brain and other organs. So, it causes a serious and often fatal diseases. You may be familiar with this disease, that is amoeboic dysentery or amoebiasis. So here is a picture of Entamoeba histolytica. So coming to the structure of Entamoeba histolytica, so this exists in two distinct forms. First one, magna or trophozoid form, and second one, that is minuta or precystic form. So trophozoid form, the mature parasite or active and motile adult is known as trophozoid. So this trophozoids of Entamoeba histolytica are large, hence the name what we commonly call magna, usually about 20 to 30 microns in diameter. The trophozoic form of Entamoeba histolytica develops from small minuta form. It enters into the mucosa and submucosa layers of the intestinal wall by dissolving its tissues. It makes small wounds in the intestinal lining which later develop into ulcers. After reaching into the intestinal tissue, that will inject the RBCs and grow up to 60 microns in size. The presence of RBC in the food vacuoles and the special type of nucleus, what we call it as cartwheel shaped nucleus, are the characteristic features of the trophozoids of Entamoeba histolytica. So you can see the picture of trophozoid stage. The cytoplasm of trophozoid is differentiated into two distinct portions, that is outer ectoplasm and inner endoplasm. The ectoplasm is clear, non-granular and hyaline, while the endoplasm is granular and fluid-like. You can see in the picture. The pseudopodium, which is a locomotory organ, may be short, broad and rounded, long and finger-like, mainly composed of ectoplasm. The endoplasm contains the nucleus and food vacuoles containing tissue fragments, erythrocytes, leukocytes and ingested bacteria, etc. The nucleus is rounded 4 to 6 microns in diameter and is vesicular. So, the nucleus of tropozoid, it is composed of a delicate membrane, small peripheral chromatin granules and centrally located small dot-like nucleolus or endosome or karyosome and chromatin granules arranged in spoke-like striations. So, here in this picture you can see that the nucleolus is surrounded by an indefinite clear area called halo. The presence of RBCs in the food vacuoles is an important characteristic of this parasite, and this has the capacity of ingesting RBC, which is not seen in any other intestinal amoebae of man. Contractile vacuole is entirely absent, which is a main characteristic of protozoans, because Entamoeba histolytica lives in the environment which is isotonic. The osmotic concentration of its body remains equal to its surroundings and hence no water enters in its body by osmosis. Therefore, there is no need of contractile vacuole. So, you already know that the function of contractile vacuole, that is osmoregulation. So, minuta, it is a precystic form which is smaller, spherical, non-feeding, non-motile and non-pathogenic. So, here in this picture you can see the minuta or precystic form. So, it measures about 7 to 10 microns in diameter and resembles the tropozoid form in its structure, except that it is smaller in size, having no pseudopodia and contractile vacuole. It lives only in the lumen of intestine and rarely found in the tissues. So, it undergo encystation, that means the cyst wall, and helps in the transmission of the parasite from one host to another. So, coming to the life cycle of Entamoeba histolytica, you already know about this. It is a monogenetic. Mono means single, genos means race. That is, its life cycle is completed in one host, the host being the man. So, in this picture, you can clearly see the life cycle of Entamoeba histolytica. So, it's an important character of an protozoa, that is encystment, that is a protective wall which created on the outermost layer. So, encystment, the precystic forms under certain circumstances remain small, that is 7 to 10 microns in diameter, and live only in the intestinal lumen. So, they undergo encystment, but before encystment, the parasites round up, eliminate food vacuoles and accumulate considerable amount of food materials in the form of glycogen and black rod-like chromatoid granules. Each parasite secretes a thin, rounded, resistant, colorless and transparent cyst wall around it, and the process we call it as encystment. Here is the picture of, first it is a picture which shows about tropozoid stage, B it is a minute or precystic, second that is uninucleate cyst, second one that is a binucleate cyst, and third one quadrinucleated cyst. the cyst of Entamoeba histolytica vary in size from 10 to 20 microns in diameter. Its cytoplasm is clear and each cyst is mononucleate at this stage. The nucleus of the cyst divides twice so that each cyst now becomes tetranucleate. At this stage, the cyst is infective to a new host. Encysted forms passed out with fecal matter of the host. The presence of chromatoid bodies is a characteristic feature of the cyst of Entamoeba histolytica. They occur either singly or in the multiples of 2 or more. So next event, that is transfer to new host. The infective cyst remained viable for a considerable length of time outside the human intestine if environmental conditions are favorable. So infection of fresh human host takes place by swallowing the infective cyst with contaminated food and drinks. Contamination of food and drink, it is brought about by houseflies, cockroaches and food handlers. Through contaminated food or drinks, the infective cysts passed into the lower portion of the small intestine of the new host. Here is the picture where exactly you can see the typical feco-oral life cycle of Entamoeba histolytica. So now the important phase, that is excystment. The word has its meaning, that is excystment. In the process of excistation, a single tetranucleate amoeba, that what we call it as metacystic form, emerges from a cyst through a minute pore in the cyst wall. The tetranucleate metacystic form produces a new generation of tropozoites by a diverse series of nuclear and cytoplasmic division, which results in the production of 8 uninucleate amoeba, and these are called as metacystic tropozoites. No sexual phenomenon have been observed during these changes. The metacystic tropozoites feed on the contents of the intestine and grow in the size to form the tropozoites of the next generation. The tropozoites stay in the lumen of the intestine for a particular period when they may attack the wall of the intestine and they again start the life cycle. Coming to the second part, that is the pathogenicity or pathogenic effects of Entamoeba histolytica. So very serious problem, that is amoeboic dysentery, and along with that obsession in liver, lungs and brain, and also you can find non-dysenteric infections too. First one, important pathogenicity, that is amoeboic dysentery. Entamoeba histolytica secretes a tissue dissolving enzyme that destroys the epithelial lining of the colon and causes its necrosis and forms the small wounds or abscession, which later become flask-shaped bleeding ulcers. The cavity of these ulcers is generally filled with the mucus, bacteria, amoeba and cell debris. The abscesses pour their content into the lumen of the intestine. The ulceration of the colon may produce severe dysentery. In amoeboic dysentery, the stools are acidic and contain pure blood and mucus, in which swarms of amoeba and blood corpuscles, they are usually present. So it also causes obsessions in liver, lung and brain. Entamoeba histolytica may be drawn into the portal circulation and carried to the liver. In liver, the parasites settle, attack the liver tissue and form abscess, that means the small wounds. The patient has pain in liver region, fever and high leukocyte number, a condition referred to as amoeboic hepatitis. Lung abscesses are usually caused by direct extension from a liver abscess through the diaphragm. The lung abscesses usually rupture into a bronchial tube and discharge a brown mucoid material which is coughed out with the sputum. Sometimes the parasite also forms abscessions in the brain. So some of the non-dysenteric infection which is caused by, that is, Entamoeba histolytica are abdominal pain, nausea, bowel irregularity with headaches, fatigue and nervousness in the minority of cases. If the severity it is very low. Coming to the therapy or treatment of Entamoeba histolytica, for acute or subacute dysentery, the infection of emetic are given. Certain antibiotics such as fumagillin, terramycin, erythromycin and aureomycin are more effective and may be given orally. For eradication of intestinal infection or in chronic cases, certain arsenic compounds, that is carbazone, thiocarbazone and vioform, and number of iodine compounds, that is etrin, diodipquine and vioform are very active. For amoebosis of liver or lungs, that is chloroquine is quite effective. The most significant advancement in the treatment of amoeboic dysentery is the use of metronidazole and tinidazole as both luminal and tissue amoebicide. Coming to the prevention of infection which is caused by amoeboic, that is Entamoeba histolytica, that is sanitary disposal of fecal matter, perfect sanitation and protection of water and vegetable from pollution, washing of hands with antiseptic soap and water before touching the food, cleanliness in preparing the food, protection of food and drinks from houseflies, cockroaches, etc. Raw and improperly washed and cooked vegetables should be provided. Thank you.